So last part from the topic, molecular basis of inheritance. So last term is the Human Genome Project. It is also called as the HGP. It means Human Genome Project. Okay. So what is meant by the Human Genome Project? So this study goes deeply in the field of the genetics. Okay. So characters, inheritance characters, genetical characters, deeply studied from the human beings. Okay, so which characters are responsible for the particular formation of the characters in the human being? Okay, so it's a deep study about the genetical character. That is the HGP, Human Genome Project. So particular genome is responsible for the particular characters. Okay, so this is the study of the genetical characters from the human beings. Okay, so in this term, the Human Genome Project deals with the study of. So what actually we have to study in this Human Genome Project? So we have to study the entire DNA in the haploid set of the chromosomes. The entire DNA in the haploid set of the chromosome. Haploid means it's a single number of the chromosomes. Okay, so that is the haploid set of chromosome of an organism is called as the genome. So actually genome is nothing but it's a set, that is the haploid set of the chromosome, for example, from the human beings. Okay, so haploid set of the chromosome of an organism is called as the genome. Okay, but when we get the chromosomes in the diploid conditions, means that the pairs, okay, so pairs will be comes together to form the haploid conditions. Okay, so that will be the haploid to diploid condition, right? In human genome, the DNA is packed in 23 chromosomes. You know, one person, for example, male as well as the female. Okay, so DNA is packed in the 23 chromosomes. The human genome contains about 3 is to 3 into 10 is to 9 BP, base pairings. Okay, so that means with the calculation near about 3 into 10 is to 9 BP. That is the base pairs. Okay. The Human Genome Project is discovered, or it will be deeply studied in the year 1990 to 2003. So this duration, this period is required for the study of the details human genome. So search starts from the 1990 or it will be ends with the 2003, and it was the first mega project for the sequencing of the nucleotides and mapping of all the genes in the human genome. Okay. HGP also coordinated by the United States Department of Energy and the National Institute of the Health. So it will be deals with the US Department of Energy and the National Institute of the Health. The goals of the HGP is what are the goals? So behind it, to study the human genome, what are the goals? So this Human Genome Project involved into two major approaches. So what are the two major approaches? So first approach is to express the sequence tags. So that is in short form, we call it the ESTs. ESTs means express sequence tags. The express sequence tag is focused on identifying all the genes that are expressed as a RNA. Okay, so they identify all genes that express as RNA. So what may be the relation between the genes with the RNA? They can firstly identify. So this is the first approach in the Human Genome Project. What about the second approach? There is a second approach is the sequence annotations. Okay, what do I mean by the sequence annotation? In the sequence annotation, the sequencing whole set of genome. Okay, so there is a one set, two sets, third set, like that. The whole set of the genome containing all the coding. Just we studied in the previous lecture, codons and coding. Okay, so all the coding and non-coding sequences, and later assigning different regions in the sequence with functions. So whatever may be there is a one set of the code, it will be having the particular function, it carry the particular characters. So all the codes are studied in the sequentially for the regions in the sequence with their functions, properly functions. Okay, so these are the two approaches behind the human genome projects. Okay, so we can see here there is a promoters. So the same thing is there. There is a promoters, then there is a exons is there. There is a end. There is a end position is here. So starting from this codons and ends with the these regions. Okay, so regulation sequences are here. Then actually the procedure going on in the HGP. So what scientists do in this project, the Human Genome Project. So procedure is like this. First, in the first step, what happened? In the first step, there is an isolation DNA from a cell. So in this step, we have to isolate the DNA from a particular cell of the organism. For example, human, we have the example human is there. So from the human cell, there is a isolation of the DNA. We have to remove the DNA from the human cell and it will be get isolated. Okay, so isolation must be required. Then after the isolation, we convert into random fragments. So actually the DNA is get into fragments with the help of the scissor, but we cannot be use the hard scissors, metal scissors. Why? Because the cell is a very small unit and in that cell there is a DNA. So how we can cut the DNA into small pieces? So there is a technique to cut the DNA into small pieces with the help of the enzymes. Okay, so that enzymes helps to get fragments into various pieces from the DNA. Okay, so it will be get fragmented DNA. Then another step is clone in a host. Clone in a host. So that host is required for the DNA. So we can use the host as well as bacteria and yeast. Okay, we can take any one bacteria and yeast using vector. For example, BAC and YAC yeast. Okay, BAC bacteria and yeast for amplification. Means there is a sequencing, study of the sequencing the characters, sequencing the codons. That is called as the amplifications. Okay. The sequencing of the fragments using automated DNA sequences means whatever maybe the sequences are there, it can be studied. Means some specific sequences are there in the DNA. So we have to study easily using the Frederick Sanger's method. So Frederick Sanger method it will use the study of the DNA sequences. So we can use that, the DNA sequence methods. After that, arrange the sequences based on overlapping the regions. So artificially, we can also overlap the sequences in the various particular regions. Okay. And then after arranging the bases in a particular region, then alignment of sequences, we can alignments. Okay. So there is a scattered sequences, scattered codons are there. And then there is the arrangement of the sequences in a particular manner. Okay. And using the computer programs. So we can use the computer program and in that there is arranging the sequences and according to that sequences, we also arrange the DNA sequence. Okay. So this is the procedure going on in the Human Genome Project. So after this completion of the process, we can study a particular character of the human genes. Okay. So here two things are important. BAC means bacterial artificial chromosomes we can make, and VAC, YAC means yeast artificial chromosome. Okay, remember this YAC and BAC. Then the Human Genome Project procedure we will there is already discussed. Then it will be carried with the isolation from this chromosome. Okay, isolation from the chromosome, from the nucleus of the cell, from the human being, and then it will be isolated the DNA, and the DNA get analyzed in the computer system with sequencing the various bases. Okay, so this is the technique. We can also get with the help from there is the genome samplers and introducing the sequencing methods is also there in the computer systems also studied and arranging the sequences. Okay. Then the Human Genome Project is studied by the Frederick Sanger. So Frederick Sanger was one of the scientists who studied deeply the HGP process. Sanger has also developed method of the sequencing of amino acids in the proteins. So he discovered the method to sequence of amino acids. So actually, naturally, the sequences are there, no doubt, but the particular sequence must be seen in the DNA, and it will be studied with the help of the protein. So that in the protein there is a sequencing of amino acids. Why? Because the proteins are act as the amino acids. Okay. So DNA is converted to random fragments as there are technical limitations in sequencing very long pieces of the DNA. Okay, so he has some difficulties. Okay, that difficulty is fragments as there are the technical limitation in sequencing very long pieces. Okay, so we have to cut the DNA into various pieces, but he was not successful to cut the DNA into many pieces. So okay, only the long pieces is there. HGP was closely associated with the bioinformatics. Okay, so it is a relation between the Human Genome Project and the bioinformatics is very close. Okay, so what actually we have to done in the bioinformatics? So there is a complete study of the human genetic characters, genetic characters with their sequences of the DNA. Actually, what is the type of the DNA particular sequence? Okay, so each and every person has a particular DNA sequences. Okay, so no similar DNA sequences between the two persons. Okay, it has a identity. We can say it's the identity of the human beings. The bioinformatics also deals with the application of the computer science and the information technology to the field of biology and the medicine. So we can get with the help of the technology, computer technology, to analyze the DNA study. Okay. The Human Genome Project also carry some important information while studying the genetical characteristics of the human being with the help of the DNA. So it is called as the genetic mapping. So genes can be mapped on the same fragments. Okay, so there is a selection of the fragment of DNA and it will be get analyzed. Okay, 24 chromosomes. It means 22 autosomes and one X and another Y chromosomes. Okay, so we will get total number is the 24 chromosome. The last sequence one is chromosome 1. It may be in the study in the May 2006. Okay, so last one chromosome. Last. Genetic and physical map on the genome were generated using the information on the polymorphism of restriction endonuclease. So what is meant by the restriction endonuclease? This is the name of the enzyme, restriction endonuclease. Okay, so it will be play a very important role while the generating the information. Recognition site and some repetitive DNA sequences. Okay, so repetitive DNA sequences called as the microsatellites. Okay, so they also get help with the study of the HGP. Okay. The DNA sequencing also have been done in bacteria, in yeast. Okay, so that is the elegance. Elegance means there is the one term, that is the bacterium and the yeast, that is the Coenorbaditics, Coenorbaditics elegans. So this is the term using in the sequencing DNA in the bacteria and the yeast. A free living non-pathogenic nematodes. Okay, so this will be host and parasite, relation between the host and parasites, one of the species, okay, from the nematodes. We also get one example from the say the Drosophila melanogaster. It is one of the insects. Okay, plants, rice and Arabidopsis. Okay, so these are one of the examples, etc. are studied the details about their genetical characters and genomes. Okay, so up till now there will be the study in case of the Drosophila plants, nematodes also, Arabidopsis also, and rice also. Okay, so there is a sequencing of the DNA. So first we will get the sequence of the DNA, then we can make some changes in the DNA sequencing and to improve the varieties of the crop plants also, and also improve the varieties of the animal species. Okay, so there is a newly hybrid animals will be formed with the help of this technique. There is the information about the genetics and their characters. Okay, so this project is very helpful to change the genetic characters in the organisms. Okay, the Human Genome Project has some features. Okay, some important features which is described here. So some features are the 99.9% nucleotides, means there is a possibility, only one impossibility. Okay, so 99.9% nucleotides base are the same in all people. Nucleotide base are same, just like the atosems. Okay, so atosems are totally same, but only 0.1%, that is the 3 into 10 raised to 6 BP base pairs differences makes every individual unique. Okay, so only 1 point chance is there to we are differ from each others. Okay, with the characters, genetical characters. So that's why every individual is a unique. Okay, so no similar human being is theirs. Okay, the functions of over 50% of the discovered genes are unknown. Means some genes are there. What type of the function they perform, we cannot be know. So 50% of the discovered genes are unknowns. We cannot be know the particular functions. What may be the take part in the process of that expression character or not. Okay. The chromosomes, one has most genes. Okay, so chromosome one has most genes, so it will be near about 2968, and the Y has the fewest, only 231. So the maximum number from the chromosomes 1. Okay, that will be there. The less than 2% of the genome codes for the protein. So it will be reserved, less than 2% reserved for the protein codes. Okay, so it will be the only 2% possibility. This will be the chromosome arranging all the genes, then genes on the, there is the chromosomes, and there is a DNA. Okay, so sequencing the DNA in the proper manner with the codes. Okay, so this will be the silent features of the Human Genome Project. Then second term is the DNA fingerprinting. Okay, so DNA fingerprinting is nothing but it's a type of the analysis. Okay, it is also a type of analysis of the genetical characters. But the difference is that in the HGP, there is a complete analysis of the chromosome with their character, with their sequences, with their base pairs. But in the DNA fingerprinting, we plot a graph. We plot a graph just like our identity, just like our fingerprints. Okay, so fingerprints can be analyzed just like our DNA also analyzed with the plotting a graph. So a graph will be obtained with the help of this technique, DNA fingerprinting technique, and there is a profile of the particular human beings will be made. So that profile is not similar to the another person, means self-identity. So in future, it may be a reliable identity of the human being. It's not our identity card, our employment card, our I cards. Okay, so reliable identity in the future, only the DNA profile. What type of the profile have a particular human beings. Okay, so it will be get printed and it will be used for the various purposes. Okay, बऱ्याच कारणासाठी त्या ठिकाणी ती वापरली जाईल भविष्यामध्ये, म्हणजे तुमची DNA analyze त्या ठिकाणी प्रोफाइल म्हणतो आपण. Okay, so on the basis of the DNA profile, we also get information about the previous history of the human being, generations of the human being, what type of the diseases are carried by the generation to generations, means deep and detailed study about our DNA, which will be studied with the help of the DNA fingerprinting. Okay, so that is the case history of the human being, genetical case history. Okay, so what actually you have to done in the DNA fingerprinting? The DNA fingerprinting is a profiling, also called, it is the technique to identify the similarities and differences of the DNA fragments of the two individuals. For example, we have the two samples from the human. Okay, so there are the two samples. So what type of the similarity is there and what type of the differences is there? So in the HGP project, so there is the similarity in the particular genes or particular chromosome, but there is only 2% dissimilarity, and on the basis of that 2% dissimilarity, it's a unique character of the human being. So 2% made the unique characters in the human beings. Okay, so that will be the, we have to study the differences of the DNA fragments of individuals. Okay, so this DNA fingerprinting analysis firstly discovered by one of the scientists, that is the Alec Jeffreys, in the year 1985. So this is the photograph of Alec Jeffreys, which will be most contribution in the DNA fingerprinting technique. Okay. The DNA carries some non-coding repetitive sequences. Non-coding, it not be get coded and repeated, okay, in the DNA sequences. So they cannot be have some codings, only just they repeated, they say make copies, and they maybe carry on the two strands of the DNA. Okay, the number of repeats in specific from person to person. So they get the person to person specifically and duplicated, and again carry in the next cell, again next cell, whatever may be. Okay, so there is a division of the cells, it can be get copied and comes in the generation to generations. Okay, so there is a no change. Okay, for example, the person one having repetition near about 8 times. Okay, 8-time repetition is there, and the sequence of the base pairs are CTAGA. Okay, sequence of the CTAGA. So it will be get copied and repeated 8 times. Okay, CTAGA. Here also repetition, CTAGA. So 8 times will be repetition is there, and some only 2% chance to get sequences of the DNA base pairs. Okay, in person 2 what happened? In person 2 also CTAGA, it will be get also copied in the repeating copied, next is the 8 times. Okay, so person third is also get copied, but is the 9 repeat times. Okay, so 9 times it will be get replaced, CTAGA. Okay, then again here will be the combination, same combination will be, we can copy CTAGA, CTAGA. Okay, so like this, there is a repetition and repetition of the same codes, same genetical characters with the codes, coding the characters. Okay, so like this, there is a person 1, person 2, and person 3. It will be differences with repeating their characters with the sequencing of the genomes. Okay. The DNA fingerprinting basis. Okay, so what is the criteria? What is the base for the DNA fingerprinting? There are the repetitive DNA. It can be separated from the bulk. Okay, for example, the characters are there, the repeated characters are there, so we have to firstly separate the characters. Repeated characters firstly get separated from the DNA. So there is one of the method that is called as the density gradient centrifugations. Okay, that method is called as the density gradient centrifugations. So actually what happen in this technique, the uniformly DNA will be get collected. Okay, and then there is a density gradient with the help of the bulk genomic DNA as a different peaks during the density gradient configuration. Okay, so it will be get configured. Okay, then there will be the force, that is the centrifugation force, and with the help of the centrifugation force, it will be get separated in the gradient manners. Okay, there will be the separation in the gradient manners. So satellite, bands, and major peaks. Okay, so satellites, it will be get arranged here, means the end of the chromosome. Satellite means what? There is a end of the chromosome. Okay, so whatever may be the configuration of the particular gene, it will be get repeated, it will be centrifuged. Okay, it will be centrifuged, and then there is a sequencing of the particular bands. Okay, so there will be the development of the particular band, that is the satellite bands. Okay, and this is the major peaks. So measuring and satellites bank arranged here after the centrifugation process. The bulk DNA form a major peak, and the small peaks are called as the satellite DNA. Okay, so satellite DNA means what? The bulk DNA forms a major peak. Okay, So it will be form a major peak. So that is called as satellite DNA. Correct. Okay. Next, the DNA fingerprinting basis also has a microsatellite. Okay, so microsatellites, it is a very small, it's a small in length. Okay, microsatellite. The short tandem repeats, STR, we can call it in short STR, short tandem repeats. Okay, the satellite DNA is classified. Okay, we have again 10 minutes, no problem. Satellite DNA is classified as microsatellites, mini satellites, etc. So there are the two types of the satellites connecting with the chromosomes, that is the micro and mini. Based on the base composition, A, T, adenine, thymine, rich or GC, rich. Okay, guanine, cytosine. The length of the segments and number of the repetitive units are there. Okay. A DNA sequence which is tandemly repeated in the many copy numbers is called as the variable numbers. That is the tandem repeats, VNTR. Okay, VNTR means variable number tandem repeats. So that is the this term also asked in the examination for the MCQ. What is meant by the VNTR or VNTR means dash dash. Okay, so VNTR means variable number tandem repeats. Okay, then we can also see here there is a graph. So repeatedly 8 times, then repeatedly 20 times. Okay, so here there is a calculation of the same process of the DNA fingerprinting. Okay, and then it will be get analyzed and plotting a graphs. Okay, so these techniques which will be provides the information about